this kind of product is produced in an open system. It's really healthy, but to be honest, it's really bad taste. You know, you can tell it's not really good, but it's healthy, so it's fine. But also we can produce more advanced byproduct like 5-coeritin. 5-coeritin is a stain that probably you use in your lab to stain mammalian cells. So this is a product from a red microalgae that we are working now, and I'm going to show you later this work. Carbohydrate. Okay, basically it's starch and exopolysaccharide production. Here we got some example of this one. This is like our bag here in Wales. They produce in this kind of bag for the starch from potatoes, but we can produce from microalgae as well. For that reason, they are biodegradable. But also we can produce more sophisticated products like this brand. It's called Alwar. It's a huge company called Frutaron, and they use this red microalgae to produce this cream. They have different activity, anti-aging, anti-everything. So it's really nice products. Well, our work. This is part of our webpage in the Center for Sustainable Aquatic Research. We are located here in the Wallace Building. Behind the Wallace Building, we have a really nice facilities for aquaculture activities. We got professional trained people, different kind of background, biologists, aquaculturists. So, we have one of the largest facilities in the UK for microalgae production and for aquaculture research. We have several projects in design from ocean acidification until energy from algae, more than 15 different projects. And we got as well large experts in microalgae production. This is a photo of our model, it's called Craig. This is a really nice guy. Here I'm going to show you, if we can, I don't know. This is a video. No, you can.
The link underneath.
Well, never. Well, I'm going to explain. This is our biofence. In this trial, our target microalgae was Porphyrium purpureum. We bought this microalgae from the CCAP. That's microalgae bank in Scotland. We bought this microalgae, and our goal was to produce in semi-continuous culture, because normally they produce in batch culture. So they put all the nutrients in the media and they grow until the end of the cure and then harvest. But our goal is to try to produce this microalgae in a semi-continuous culture. Another goal is the production of EPS. It is exopolysaccharides production from the supernatant, and the production of 5-coeritin as well from the biomass. So this is a really interesting algae because you can use the biomass and you can use the supernatant as well. So you use all the stuff from the microalgae. These are two biofens, 6 liters each, and we grow one in semi-continuous and one in batch culture at the same time. First, we did several trials in indoor condition, in a small volume, 20 liters, in batch cultures. So these are, for example, the parameter in the harvest time, at the end of the cure, day 20 roughly, and then you can see the pool composition of this algae measured by FTIR. So as you see here, there are an increase in proteins that is concordant with the exponential phase, because they are growing, so they are building blocks and they are using proteins in order to divide and to produce more life. Also, we have an increase in lipids as well, the orange line, because when they are dividing, they use polyunsaturated fatty acid to produce cell membrane and the stuff. And after, when they reach the stationary phase, we have a huge drop in protein production, and we have a huge increase in carbohydrate. The the strategy of this species specifically is when they reach the stationary phase. It means that when they are nutrient depletion or when they are stressed, some kind of environmental stress, they release this sugar to the medium. So after that, we can extract, we can separate this this medium from the biomass, and we can obtain the EPS. So, for example, we measure there the carbohydrate production was more than 1 gram per liter at the end of this group. In that trial, we obtained that it's feasible to grow in a small scale until 80 liters. In back, they show good growth rate, 0.5 division per day. It's feasible to harvest with membrane technology. We checked this one first in a small scale because after that we need to scale up to the biofence. And this one is feasible to harvest with membrane technology because it's when they grow until the stationary phase, the culture is really gloopy, really gloopy. So we are not sure if we can harvest, but now we know that we can harvest this with membrane technology. Our maximum carbohydrate production was around 1.5 grams per liter, and our maximum theoretical production of phycobilirubin protein, that is phycoerythrin, was between 20 to 50 milligrams per liter. So then we scaled up to this outdoor condition in a greenhouse behind the Wallace building, and then you see the blue line is the indoor growth in cardboard, and the red is semi-continuous culture, and the blue one is batch culture. Here we have the environmental parameter, pH and temperature. We got in both bioframes a longer lag phase because we had problem with the pH. It was really low, was really acidic. So for that reason, we had a longer lag phase until day around 8. If we compare with the indoor condition, because they have just 2 days of lag phase. After that, they grow really well, and we are working on all the data because we are full of data. We need to analyze biochemical, we need to analyze CN, plenty of data. I have to download all the environmental data, so this is a really preliminary result. But in terms, for example, of parameter, we have temperature. It's almost the same one in both reactor. The pH is almost the same one, but we have more productivity in the semi-continuous culture. So why to use this kind of a strategy? Because then we can keep growing the algae and harvesting. It's like in theory we can harvest every 3 days because that is the duplication time. If you measure the growth rate, then you can obtain the duplication time. So in theory we can harvest every 3 days, but now we are harvesting every 7 days. So we harvest roughly the third part volume of each biofence, and it's around 200 liters. So it's really good results. And what we obtained? So we obtained this is a microphotograph of the electrospinning that we did with a colleague in the ILS and in the chemical engineering. It's called Amir, and we obtained this product. So this is a small quantity of this product. This is the EPS from the supernatant, and this is the electrospinning stuff. So we're gonna use this one for wound healing and for skin care stuff. So what we have here, so this is the raw material, the scan is the raw material, the EPS, and you will see that we have a huge peak that is carbohydrate, mainly composed for sugar monomers as a glucose, galactose, sucrose, and fructose. So after the electrospinning stuff, they mix many different things like salt, PBA, and other stuff. And our goal was to find if we got the same EPS concentration in the final product. So we got the same peak. It's good. In fact, we have almost the same percentage of carbohydrate, and we have all the different stuff like some lipids here. This is water and some protein. But we got this carbohydrate, so it's really good. So this is a microphotograph photograph of this film, and now we're going to do a really huge one in order to try to discover if we can use for other kind of applications. So this is the EBS, who will start from the SuperNet. Another good result is the phycoerythrin, that is this one. It's really expensive, this phycoerythrin. Usually it costs around 150 pounds, 100 milligrams, so probably you have here more than 150 pounds here. So we obtained this one as well, but from the biomass. The colleagues from the chemical engineering, Magru, Michael, and all the colleagues, they work with membrane technology, and in this experiment they test different pore size in order to fractionate these molecules. And we observe in this we use a spectrophotometer to check the intensity of the absorbance. We have a huge peak in 545 nanometers, and the big one, the green line, the big one is with the 10 kilodalton membrane. So we can retain all the phycoerythrin in that portion. We need to do more experiment on that, but we think that with this membrane we can fractionate this product and we can obtain this product. As you will see, the red intake is really pinky, and the pigment is really bad. So probably this is the right membrane. This is a photograph from disrupted cell, and this is the final after visit. So then we do the fractionation with the membrane. It's a really good result. So, as a conclusion, these microalgae are proper biofactories, so you can use all the cells in order to produce different kinds of products. It's feasible to use the biomass and the medicine as well to extract the products. Microalgae can grow in Welsh location, but the production must be focused on high value metabolism, because we cannot compete with Spain, Israel, and Australia to produce oil or other kind of stuff. We must focus on really high expensive molecules. Currently we are focused on ETS and phycoerythrin production, but in the near future we will want to use another kind of strain that is a dinoflagellant, and with this kind of strain we can produce other kind of high value metabolites, such as macrolides. Macrolides are the raw material for antibiotic stuff. So maybe later, if we want to do some kind of collaboration with us to check our macrolides, we can produce as well luteophenol with this kind of species. Luteophenols have antifungal properties, and even we can produce DMS with this the new species that we are starting to grow in the near future. We need to explore local microalgae strains such as Micrococcus pluvalis. In fact, when I did my internship here in 2010, I isolated one Nematococcus from Mambuts, and we had the master photo, and we want to start to grow this one in the near experiment that we have with the people with chemical engineering. And we need to establish new collaboration between different colleges. If we want to do science, we need to establish this kind of collaboration. So if you want any kind, if you have any kind of comment or question to me, is